Hello everyone, a very warm welcome to the development of the inner ear. Before going to the development of the inner ear, we see what are the various part of external ear, middle ear and inner ear. So let's see this external ear mainly constitute of pinna and external auditory canal, which is also called as external acoustic meters or ear canal. You can see this is the pinna, which is also called as auricle, and this a type of a tubular structure with a hollow lumen that is called as external acoustic meters or ear canal. And this pinna itself constitute of a very small cartilages, and also this the physical appearance give various names like helix, which are the broad upper area, that is helix, and here the shaded region in this just beneath the helix, this broad thickened area of the helix, just beneath that there is a scapha, and also beneath that there is a triangular structure. This triangular structure is called as a triangular fossa. And next you can see there is a reflection type of structure that is called as anti-helix. Just beneath this helical structure, you can see this is the anti-helix, and this smooth area near the opening of this auditory or ear canal, that is called as the concha, and this smooth structure is now called as auricular lobule. So all these part are the structures within the pinna or auricle. And next we see the part of the middle ear. Middle ear constitute of auditory ossicles that are arranged within tympanic cavity. This auditory ossicle constitute of the three tiny bones like the malleus and incus and stapes. You can see this malleus is connected to the tympanic membrane or eardrum at one end, and adjacent to it there is an incus, and this incus is externally attached to this malleus, and internally that is attached with the stapes, and the step is attached to the vestibule of this uh, this vestibular apparatus within the inner ear. And next we see in the ear, in the inner ear, you can see there is cochlea vestibular apparatus. Cochlea is these coil structures, and this vestibular apparatus include semicircular canals and article. Article is present somewhere here, and the saccule is present here. This saccule is connected to the cochlea through this ductus reunions. Ductus reunions will connect this saccule to this cochlear structure, cochlear apparatus, and this is also the part of the inner ear. You can see in the structure of the inner ear there is a semicircular canal, and the semicircular canal is itself constitute of the three semicircular canal. First is there is a posterior semicircular canal, anterior semicircular canal, and also there is a lateral one. And just before opening into the auricle of this vestibule, there is a small expansion at each end of the semicircular canal, and this is called as ampulla. And also there is a endolymphatic sac, and this endolymphatic sac is connected to the vestibule through the endolymphatic duct, and this is the cochlear duct, and this cochlear duct connect the saccule to the cochlea. So all this structure comes under the inner ear. So let's begin with the development of the inner ear. Generally, all the derivatives of the inner ear arise from the ectoderm. What happens that during the third week of the development, the surface ectoderm just adjacent to the rhombencephalon that will thickens, and that thickening will form the otic placode or otic disc, and that we can see here in the you know head region of the embryo. You can see there is the invagination, and this invagination is a otic pit. What happens that this otic placode has cells, you know um, epithelial cells, and that will invaginate during the third and the fourth week of development, and that invagination will form the pit like structure in the cranial part of the embryo, that is called as the otic pit. You can see these are the cells of the otic placode that are being invaginated into this structure, giving rise to the otic pits. Next, we see that third week, during third week and the fourth week, this otic placode will form the otic pit through the gradual invagination of the ectodermal cells. And later on, what happens that this is the surface ectoderm, and this surface ectoderm will thicken in comparison to the adjacent ectoderm, surface ectoderm. This thickening will form the otic placode, and this otic placode, what happens that their cell will invaginate, and that will form the structure like this, and this structure is called as the otic pit. And this is a otic placode, and this is the otic pit, and this pit formation occur during this third and the fourth week, and this also occurred through you know near to this time. And what happens that this is the surface ectoderm, and later on this surface ectoderm, this will fuse to adjacent site, and this fusion lead to the formation of the otic vesicle or otic cyst. Now this is called as the otic vesicle or otic cyst, and this otic vesicle or otic cyst is connected with this surface ectoderm with the help of a stem, and this stem later on get obliterated, and this otic vesicle will be completely detached from the surface ectoderm. So these things are seen here, and also we can see that there in the ventral otocyst, that there is a dorsal side and the ventral side. So in the ventral region, there is a certain young neurons, and these young neurons that are initially presented, yes, and these undergo delamination, and this delamination later on form the vestibulocochlear ganglion. So all these things happen here during the third and the fourth week. You can see there is the otic pit formation, and this is the otic placode, cells of the otic placode, and you can see there is the delaminating young neurons which later on form the vestibulocochlear ganglions. And all these things happen during the 25th day. You can see otic placode here, and here you can see there is the otic vesicles, and they are connected to the surface ectoderm with the help of the stem. And you can see there is the programmed cell death of this stem, which is connecting the otic vesicle to this surface ectoderm. And along with this otic vesicle formation, you can see there is the delamination of the young neurons, and that delamination is forming the acoustic or vestibulocochlear ganglion side by side. So you can see here. Next we see that by the 28th days, what happens that this otic vesicle, we saw this otic vesicle which was initially connected to the surface ectoderm. After getting detached from this surface ectoderm, what happens there is this is the dorsal region, and this is the ventral region. And you see this is the otic uh, vesicle. And by the day 28, the dorsomedial region, this dorsomedial region of this otic vesicle elongate, that will elongate and form this into lymphatic appendages. Yes. And this body of this otic vesicle, that otic vesicle, the body of this otic vesicle will form superiorly, it will form the pars superior, and inferiorly towards the ventral region. This is the ventral region and this is the dorsal region, and towards the ventral region it will form the pars inferior. Yes, you can see this is the body of this otic vesicle, and that is differentiating into this expanded superior, pars superior, and tapered. That is the tapered end. Yes, tapered towards the tapered end ventrally, that will form the pars inferior. So let's appreciate from the figure. Here you can see this is the otic vesicle, and this otic vesicle by the day 28, by the day 28, you can see there is the elongation of this dorsomedial region of the otic vesicle, which is forming the endolymphatic appendages. And also you can see the body, the region of the body that will form the pars superior, which is slightly expanded region. You know, this is the slightly expanded, and you can see the slightly tapered end, which is pars inferior. So these things are being differentiated from this otic vesicle, which is initially found from the thickening of the surface ectoderm just adjacent to the rhombencephalon. So next we see the, you know, this endolymphatic appendages, that we saw that there is, you know, structure formation like this, that might not be similar to that, but you can see that this is the pars superior, slightly expanded region, and this is the tapered end. This tapered end is called as the pars inferior, and this dorsally located region, this is the endolymphatic appendages. Yes, and this endolymphatic appendages, you see here that this distally that will form the endolymphatic sac, and the remaining portion will form the endolymphatic duct. This endolymphatic duct will connect this endolymphatic sac to this pars superior. You can see this distal region will form the sac, and this will form the duct, and that duct will connect this to the pars superior initially. And next you see that during the fifth week, the ventral tip of the pars inferior, you can see we had drawn, this is the endolymphatic structure. Here there is a sac-like structure that is formed from the endolymphatic appendages, and there is the expanded pars superior, and next there is a tapering uh, pars inferior, and this pars inferior that will elongate more and more. The cells of this pars inferior will elongate more and more, and that elongation later on will form the coil, and this coiling plus this elongation that will lead to the formation of the cochlear structure, and that is the cochlear duct here and the cochlea itself here. So these things are found from the pars inferior. So in the picture you can appreciate like this here, this endolymphatic appendages, you can see that is forming the endolymphatic sac, and next that is forming the endolymphatic duct, and the pars superior is connected to this endolymphatic sac through this endolymphatic duct, and this pars inferior you can see here. Next you see this cochlear duct, which we discussed earlier, that this elongation from the pars inferior that is forming the cochlear duct and this cochlea itself. Next we see that the pars superior, that will flatten dorso laterally, that begins to flatten dorsolaterally, and this flattening or this uh, development, this growth, dorsolateral growth of the superior pars superior lead to the formation of the semicircular canals. Three semicircular canals, that is the posterior, anterior, and lateral one. So this dorsolateral expansion of this pars superior, that is forming the posterior, anterior, and this lateral semicircular canal, and also this is happening during the late fifth week. And next you see that there is also the formation of the utricle and saccule. This utricle and saccule, these are also forming from the, this is the flattened part of the pars, um, that is the pars superior, and the remaining portion here, that will differentiate to form the utricle and the saccule. Now this saccule is connected to the cochlear region with the help of ductus reunions, that is presented here with a narrow structure. This narrow structure is connecting the saccule with the cochlea. And also we see that there is a, that semicircular canal that is connected with the vestibule here, vestibule, yes. And the both end of this canal is opening into the vestibule. Before the opening, the one of the end of the semicircular canal, that will form the small expansion at each region, and that small expansion is called as ampulla, that is represented here. You can see that ampulla. And also you can see there during the seventh week, the shells of this cochlear duct, this cochlear duct will form the spiral organ of the corti, the hair cells, that will not exist throughout in the adulthood, that will differentiate into the spiral organ of the corti hair cells. Next we see that there is a small expansion called the ampulla that houses the sensory cells, and that formed at one end of each semicircular canal. You can see the posterior semicircular canal that is opening here in the vestibules, and before opening there is the ampulla small expansion here. And also this is the lateral semicircular canal, and it has its own ampulla. And also this is the interior semicircular canal, and it has its own ampulla. So you can see there is the three ampulla, that is the small expansion that is present at the each end of the semicircular canal, each semicircular canal. So the ampulla related to the posterior semicircular canal is called as the posterior crista ampullaris, similarly lateral crista ampullaris and anterior crista ampullaris. And next you can see the article and saccule, that will also form the saccular maculae, articular maculae, and statoacoustic ganglion, that is found from the delamination of the young neurons. So all these things are being developed in the inner ear. Next we see that the prosensory regions in the cochlea, that will form the organ of the corti, and also the saccule and article forms the maculae. You can see that article, initially it was present as article, and later on that will differentiate to form the articular maculae, and that saccule will form the saccular maculae. And all of these sensory regions, all of these sensory regions that are innervated by the statoacoustic ganglion of the vestibulocochlear nerve, and these vestibular structures are innervated by the vestibular branch, these all vestibular structures. And the Cochlear regions. The cochlear regions are organ of the corti that all are innervated by the cochlear branch. So, in the summary, let's see what we studied. That we see the development of the inner ear, and in the development of the inner ear, what happens? The surface ectoderm just adjacent to the rhombencephalon that thickens to form the otic placode, and this otic placode, its cell will invaginate. This otic placode formation occurs during the third week of the development, and the cells of this otic placode will invaginate. Yes, that invagination will lead to the formation of the otic pit, and this otic pit formation will occur during the third to the fourth week of the development. This otic pit later on, after the fusion of this surface ectoderm, that will lead to the formation of the otic vesicle, which is connected to the surface ectoderm with the help of this stem like structure. Now this is the otic vesicles, and these otic vesicles later on get separated from the surface ectoderm after the programmed cell death of the cells that are present in the stem, which is connecting this otic vesicle with the surface ectoderm. So now this otic vesicle will be completely, that will be completely separated from the surface ectoderm. Now what happens, this that this is the dorsal region and this is the ventral region. Yes, and these dorsal regions, in the dorsal regions, the cell of these otic vesicles that will elongate, that will differentiate, proliferate, what happens, that will elongate and that will form the endolymphatic appendages, and these endolymphatic appendages later on forms other structures. And what happens that this is the body of the otic vesicle. Yes, and these otic vesicles will form the expanded pars superior and the tapering pars inferior, and this superior pars superior will be differentiated to form the semicircular canals, that is posterior, anterior and the lateral. These semicircular canals will form, along with there will be the formation of the article and the saccule. And next you see the pars inferior, the cells of this pars inferior will elongate and elongate and elongate, and that will form the structure, cochlear duct like structures, and this tapered end of the otic vesicles that will coil and form the cochlea, and later on this cells of this cochlear duct that will form the sensory hair cells, that is the sensory hair cells of the organ of the corti, and all these structures are formed this from the pars inferior and the pars superior. Now, the turn of these endolymphatic appendages. This endolymphatic appendages, what happens that will form the sac-like structure that is called as the endolymphatic sac, and the lower portion of these appendages that will form the endolymphatic duct, and this endolymphatic duct will connect the expanded region of the pars superior, which later on form the vestibule, which constitute of the article and saccule, and this will be connected with this endolymphatic sac with the help of the endolymphatic duct. So all these things can be appreciated in the development of the inner ear.